Biomass cultivation for fungal leather substitute production. The characteristics of the final leather substitutes derived from fungi depend on several factors, including the type of fungal strain used, the type of growth substrates and other growth conditions, the mode of fermentation, as well as the subsequent downstream processing. Among the fungi, white rot fungi have the most desirable properties required of the starting materials for fungal-based leather substitutes. Subsequently, the fungal inoculum can be cultivated under submerged, solid-state or liquid-state conditions for efficient product recovery. Being xylophagous in nature, higher fungi practically utilize a wide variety of materials for their propagation, ranging from simple defined media to complex lignocellulosic biomass that are readily available. And it has been noted that in addition to the fermentation mode, the choice of the nutrient substrates can be used to tailor the mycelium material's properties for their use in various applications. For example, there was a significant variation in the moisture level of Trametes multicolor-derived biomaterials, with different agricultural residues as the feed substrates. Average moisture levels of 6.5% and 8.6% were recorded when grown on cotton and rapeseed straw, respectively. Solid-state fermentation, SSF. SSF has been recognized as a sustainable fungal cultivation mode, especially when the production of biomass or secondary metabolites is to be optimized. Compared to its main alternative, that is, submerged fermentation, SSF tends to be more attractive due to its higher productivity, lower water and energy consumption, minimized risk of contamination, and reduced generation of wastewater. According to Chilakamari et al., fungal growth demands less water activity, approximately 0.5, when compared to bacteria, approximately 0.9. Thus, the natural growth of fungi is better simulated under SSF conditions, leading to increased efficiency. Despite the low water activity of the SSF approach, the growth substrates usually possess adequate moisture to support microbial metabolism, and this could be in the range of the maximum water-holding capacity of the solid substrate. It has been observed that the purpose of producing pure mycelial mats, which is the starting material for fungal leather substitutes, is better served under SSF. This is important because fungi typically require direct contact with their growth substrate. Thus, they tend to be intimately tethered to their nutrient source. However, the production of fungal leather substitutes demands the use of non-woven fungal mycelial mats, which can only be harvested from the top surface of SSF or LSSF trays. Basically, a mat of fungal mycelium grows on top of a solid substrate, after which the top pure mycelium layer is carefully separated from the substrate. Furthermore, the chemical and mechanical properties of the mycelial layer can also be tailored by varying the type of solid substrate used for the fermentation. For instance, the solid-state cultivation of P. austriatus on potato dextrose broth, PDB, resulted in a higher rate of hyphal collapse, reduced hyphal size, and showed different relative concentrations in chitin, polysaccharides, lipids, and proteins when compared with the growth on cellulose, thus suggesting that the harder the substrate used, the stronger the mycelium produced. In the same study, it was shown that the fungus grown on PDB had a lower Young's modulus, higher water absorption, and a higher rate of elongation relative to the latter. SSF has been noted to be the preferred mode of mass mycelium production and is currently used by companies such as Ecovative, MycoWorks, and Mycotech for commercial purposes. Liquid-state surface fermentation, LSSF. LSSF involves the cultivation of microorganisms in a liquid medium using simple culture medium or lignocellulosic substrates, or both under static conditions. As a result, the microbe grows on the surface of a static liquid medium that contains up to 95% free water. This fermentation mode has been widely used in the production of organic acids and many other volatile compounds, and was preferred to submerged fermentation due to its economic advantage and ease of product recovery. Similar to SSF, utilizing LSSF is also effective for producing pure mycelial mats. It is believed that this approach of mycelial cultivation also gives room for tunability of the material properties of the mycelium. According to researchers, the material properties of biomass can be enhanced by the addition of porous materials, such as linen or cotton, on top of the surface, allowing the mat to penetrate the layer. Variations of this method have been demonstrated in the works of different research groups. However, the major limitation identified with this cultivation method is the poor availability of dissolved oxygen within the static liquid medium, which affects biomass proliferation. Submerged fermentation, SMF. Submerged fermentation process, also referred to as liquid fermentation, is the cultivation of microbes in liquid media containing all the nutrients necessary for the growth and metabolism of the microorganisms. SMF is considered more advantageous than SSF as it allows for easier control and flexibility of process parameters, especially in the bioreactor scale. Although this cultivation mode has been used several times for growing fungi, including mushrooms, the majority of these were done to produce other bioproducts besides fungal biomass. It is believed that the cultivation of fungal biomass under submerged conditions will result in the production of slurry or pelletized biomass that would demand laborious processing before a coherent piece of material can be obtained. Expectedly, the material obtained under SMF has been noted to possess different tactile properties in comparison to fungal leather substitutes during SSF, which have a more spongy morphology. For instance, in a study aimed at producing engineered living materials from G. lucidum, cell pellets produced from SMF were blended, homogenized, and plasticized to finally form a flat, leather-like material. Such processing was also carried out successfully in the production of fungal materials with leather-like properties under submerged conditions using bread waste as the substrate. Processing of fungal mycelium into a leather substitute. After the harvesting process, the mycelial mats are made to undergo a series of physical and chemical treatments to get them in the right aesthetics, shape, texture, and mechanical conditions required for various applications. Furthermore, the different combinations and variations of treatments currently in use are mainly aimed at improving pliability over rigidity, as well as increasing the durability and absorbent properties. These treatments vary in scale and may include adjustments of mechanical properties through techniques such as deacetylation and cross-linking of chitin, protein denaturation, material densification, and moisture content control. However, the treatment processes are still undergoing intensive research. Hence, there is currently no one-size-fits-all procedure. It is also important to note that the different processing methods of the various companies involved in the development of fungal leather substitute is their trade secret. Hence, an authoritative comparison will be difficult to make. Despite this, a general overview of the currently reported processing methods of fungal mycelium to the final leather substitute is necessary. The chemical pretreatment process has been noted to either be a mild alkali or acid treatment, and these are both synonymous with the liming pretreatment in animal skin leather, which serves to remove extraneous materials while enhancing the subsequent penetration of the tanning agents. The modification of the chitin content of the fungal mycelium has also been identified as a viable avenue for the introduction of chemical modifications, thereby enabling new properties. Deacetylation of the chitin, which is also achieved during alkali treatment, is expected to release amino groups, which will facilitate additional cross-linking sites on the chitosan backbone structure in the mycelium. In addition, the deacetylation process has also been noted to improve the antibacterial properties of fungal chitin. making the deacetylated fungus-derived materials more suited for applications such as bioactive wound dressing materials. Crosslinking of the hyphal filaments in the mycelial mats is a treatment method that is expected to enhance the stiffness and strength of the alternative material as well as improve its durability. Crosslinking can be achieved chemically by using acids, citric acid, dicarboxylic acid, polycarboxylic acid, and tricarboxylic acid, and tannins, glutaraldehyde and pyrogalols. For example, citric acid was found to be effective in cross-linking the cell walls of the adjacent hyphae via its reaction with the glucan hydroxyl groups present in the cell wall. The tanning process of fungal mycelial mats is another major treatment process that is closely related to cross-linking. It stabilizes the structure of the biomaterial, giving it a texture that might mimic that of actual leather. Tanning of fungal mats has been done using vegetable tannin at mild temperatures for prolonged periods, such as 8 days at 25 degrees Celsius. The tanning of fungal biomass in reishi production was noted to be achieved using environmentally friendly fat liquors, which are chemically modified animal and vegetable oils. After tanning, the fungal sheets may be further flattened using the wet laid technique. Hot pressing can also be introduced as the mechanical, physical, and thermal properties of the biomaterials are noted to be improved by the post-synthesis pressing temperature. It is also believed that heat pressing will facilitate the homogeneity and isotropy of the biomaterial for better functionality. In a research, cold and hot pressing were used separately in the treatment of a fungal biocomposite. However, the material property, especially the elastic modulus of the biocomposite, was recorded to be improved with hot pressing than cold pressing. Other treatment processes of the fungal biopolymer have been identified to be mainly chemical treatments, which serve to further enhance the strength of the materials. For instance, treatment with glycerol, plasticizer, and a hydrophobic bio-based binder was observed to increase the flexibility, tensile strength, and water-repellent ability of a fungal-based leather substitute. In addition, the wearability and resilience of the biogenic leather substitute to perspiration, water spotting, solvent wicking, crocking, UV exposure, and salty water have also been noted to be enhanced by hybridization with other natural or synthetic materials, such as felted polyester reinforcement or polylactic acid surface coating. The post-treatment processes are mainly to improve the aesthetics of the material, and they involve large-scale modifications of appearance, which can be achieved through techniques such as carving, imprinting, embossing, stamping, and dyeing of the mats. Filamentous fungi have been identified as remarkably rich and diverse groups of species that have established themselves as promising bioresources with a wide range of industrial applications. Products derived from this group of organisms are now considered essential building blocks for a transition towards a more sustainable future for our planet, as they facilitate a pollution-free environment while providing renewable alternatives for fossil-based resources. The production of leather alternative materials from filamentous fungi via environmentally benign processes has been noted as one of these sustainable solutions. Recent development has shown that, besides the utilization of fungus as raw materials for animal leather substitutes, the list of potential applications is exhaustive, and it includes the production of biomedical scaffolds, electronic wearables, composite construction materials, packaging, support material for electronic circuit boards, wound care material, and even the manufacture of meat alternative products. However, the potential of fungal leather substitute materials in the consumer market is still undervalued, as it is faced with a myriad of challenges, which vary from the dearth of knowledge in basic fungal biology to industrial challenges. In addition, one of the biggest challenges with deploying this technology is the inability of fungal-derived leather substitutes and other leather substitutes to attain the same material properties as animal leather. It is expected that more research and development efforts will be focused on increasing the versatility and durability of these fungal-derived products, which will consequently facilitate the introduction of mycelium materials to a wider range of consumers. In the long run, the expected development of the fungal leather substitute industry, based on utility values, will be enhanced significantly by the global shift towards environmental sustainability, the availability of low-cost agricultural residues as fungal feedstock, responsible fashion trends, and government policies and regulations.